Some flu seasons are just bad, but then there are the ones that are absolutely brutal. And the winter of 2017 to 2018, that was one of the brutal ones. It hit so hard, sending way more people to the hospital and causing more deaths than we'd seen in years. But the real story wasn't just how bad it was why. The usual explanations didn't add up, leaving scientists with a genuine mystery to solve. So let's play detective and crack this case. Just to give you a sense of the scare here, take a look at the numbers from just one health system, Johns Hopkins. In that season, flu cases hit nearly 4,600. Now compare that to the year before, which was about 3,300. That is a huge jump. This wasn't just a little uptick, it was a massive surge that set off alarm bells everywhere.
And this is where the mystery really kicks in. See, when a flu season gets this bad, scientists usually have a pretty good idea why. They've got a checklist of the likely causes. But this time, none of them fit. The virus hadn't changed in the way they expected, and the vaccine wasn't a total failure. So if it wasn't the usual suspects, what on earth was the secret culprit behind this killer season?
Okay, so let's start by examining the scene of the crime, the 2017-18 flu season itself. The virus that was running rampant was a type of influenza A called H3N2. And H3N2, well, it has a reputation for being particularly nasty, especially for older adults and young kids.
And the impact was just devastating. We're talking about a massive number of people getting sick all across the country. We saw a big spike in deaths from pneumonia and the flu, and hospitalization rates just went through the roof. Hospitals were completely overwhelmed, and public health officials were scrambling to figure out what was driving this incredibly intense wave of sickness.
Now, in any good investigation, what's the first thing you do? You round up the usual suspects. And in the world of influenza, there are two prime candidates that are almost always behind a really bad season. Let's take a look at their rap sheets. First up, we have what's called antigenic drift. Basically, this is when the virus makes tiny little changes to its genetic code. It's kind of like it's changing its disguise just enough so that our immune system, which remembers last year's version, doesn't recognize it. The second suspect is a classic vaccine mismatch. This happens when the flu strains that scientists pick for the annual vaccine just aren't a good match for the viruses that end up actually going around. But here's the twist. According to the research, neither of these prime suspects was the real killer. There was no major antigenic drift. The virus hadn't changed its disguise all that much. And while the vaccine wasn't perfect, it wasn't bad enough to explain just how severe the season was. The usual suspects had an alibi. The case had officially gone cold. With the main leads all dried up, the investigators had to bring out the big guns. They had to go beyond the surface and look deep into the virus's genetic code. They used whole genome sequencing to hunt for a hidden clue that everyone else had missed. And they found it. The real culprit wasn't those tiny little mutations. It was something much more dramatic: intra-subtype reassortment. Now, this isn't just a virus tweaking its disguise. This is like two different viruses meeting up, swapping entire parts of their genetic blueprint and creating a brand new hybrid. They're literally shuffling their genetic cards to create a whole new hand. So, how does this crazy gene swapping actually work? Well, picture this. First, two slightly different H3N2 viruses have to infect the exact same cell in someone's body at the same time. Inside that one cell, all their genetic parts get thrown together and mixed up. Then, when new viruses are being assembled, they can pop out with a brand new combination of genes, some from one parent and some from the other. And just like that, a new player has entered the game. In this table right here, this is the genetic fingerprint they uncovered. The new virus that was causing all the problems, this R3C2A2, was a hybrid. You can see it clear as day. It took a couple of key genes from one parent virus and the rest of its genes from a totally different parent. This was the smoking gun. But wait, because the plot thickens. The genetic detectives didn't just find one new culprit. Turns out they found two distinct hybrid viruses, a dangerous duo that was circulating at the same time, working together to make the season so awful. It's time to meet our perps. On the left, we have the Brute. This is that R3C.2A strain we just saw. Its new genetic combo made it a powerhouse. It could copy itself much faster and reach higher levels in the body, which led directly to people getting sicker. On the right, we have the Disguise Artist. Its trick was different. It managed to grab a gene that let it sneak past our pre-existing immunity, making it much better at infecting people who should have been protected.
And this isn't just a theory. We've got the forensic evidence to prove it. Check out these lab cultures. The white spots, or plagues, are areas where the virus has completely destroyed the cells. On the left, that's the original parent virus from the year before. But look at the right. That's the Brute. The plaques are so much bigger and more numerous. It's clear, visual proof that this new hybrid virus was way more aggressive.
So what did that mean for actual patients? This chart says it all. It shows the influenza severity score for people infected with the old virus versus the new brute, and the difference is staggering. Patients with the new hybrid virus had a severity score that was double, literally double, that of patients with the original virus. This genetic swap had very real and very severe consequences.
So the case is cracked. The mystery wasn't one culprit, but a conspiracy between two stealthy, genetically shuffled viruses. So what does this incredible discovery tell us about the flu's future playbook?
What the flu detectives found was basically a perfect storm. You had the brute making people sicker, and you had the disguise artists spreading more easily through the population. The combined force of these two strains circulating together is what made that season so brutal. And here's the scariest part. This all happened under the radar, without any of the classic warning signs that public health officials usually look for.
And that brings us to the most important lesson here. We can no longer afford to just watch for the usual suspects. This case proves that detailed genomic surveillance, constantly sequencing the genes of viruses out there, is absolutely essential. It's the only way we can catch these subtle but incredibly dangerous genetic shuffles before they explode into a full-blown public health crisis.
That 2017-18 season was a stark reminder that we are in a constant evolutionary arms race with the flu. The virus is always shuffling its genetic deck, looking for a winning hand. This discovery gave us a peek into its strategy, and it leaves us with a critical question for the future. Now that we know what it's capable of, how can we stay one step ahead?